Good afternoon. Dr. Dan Guerra here from VerEvMed Studios in the Pacific Northwest of the United States. Today, I want to get into a really interesting topic that's going to be um, laden with a lot of data and a fair amount of uploading associated with how we can interpret how this paper was put together. That is very common for papers that are published in certain journals, and it's no exception here. In fact, this particular paper is published in the journal Cell. And Cell is sort of notorious for giving you a lot of detail, both in how the experiments are conducted, a lot of data, which is also fantastic because then you get to see not only the results they want to present directly, but all the supplemental material. And then, of course, really good discussion. So that's why I chose this paper. But even more because of the topic. The topic is going to assimilate and associate various topics that we've been covering over the months here at VerEvMed. They include signal transduction pathways, they include metabolic regulation, and they are also associated quite directly with tumorigenesis. So let's not talk anymore about what it's about. Let's just do it, right? So I'm Dr. Daniel John Guerra, and there's my individual email address. You can contact me about any of these video lectures and also about our company, VerEvMed. There's the website for VerEvMed, and there is also the email address that you can contact us. So two methods to contact us via the email embedded in the company website or my personal email. Either one I will respond to. Okay, so without going into any more detail about VerEvMed, we could talk about that some other time. Hopefully you are aware of the fact that we're a company that's associated with helping people interpret, first of all, to be associated with and to find out what the primary scientific literature says on a given biomedical topic or just basic research topic in natural sciences, particularly biochemistry, but also cell biology, pharmacology, pharmaceutical sciences in general, but also vet med. And also we are a service for other kinds of people that need to accumulate data directly from the published scientific literature. That includes lawyers and patent attorneys, as well as, of course, lay people who want to know more about, let's say you've been given a recent diagnosis or you're taking a certain drug and you want to know more about it. You can contact us. We can go into literature with you. We can vet that information out, and then we can share it with you, and then you would share it with your physician and your other healthcare providers. So please contact us. We're a very vibrant company, a young company, and we'd love to have your business. These lectures are free, and they're meant to be emblematic of the kinds of depth we can get into. And the topics that we cover sometimes are from listeners and from clients and colleagues that want to know more about a given topic. And also very often it's just directly from what I might find interesting in the very current literature. So that's what we're going to do today. This is going to be talking about a very important molecular switch called mTORC. That's the target of rapamycin, and the C means complex, and how it's involved in the transcriptional control over fatty acid synthesis by altering the binding and stabilization of nascent fatty acid synthase messenger RNA intron transcripts. So I know it's a big mouthful, but some of you I know are fully aware of what I'm saying there, and those that are not, don't worry, I will go into the detail. By the way, there's my Facebook page. All of my recordings are on my Facebook page. Please go to it and you will find them. So let's not talk anymore. Let's just get going. This paper I'm going to talk about was published just about a month ago. So I'm recording this actually on the 29th of January, 2018. So this was about what? Oh, maybe, yeah, I guess 6 weeks ago. This was published in Cell. So let's get into the background. What is the general introduction I want to tell you? mTORC, okay, then that's an enzyme complex, which is basically a kinase. That means it adds phosphate to other proteins. Regulates anabolic pathways at the transcriptional level. So mTORC1 is a majority protein complex that responds to various intracellular signals, including growth factors, membrane-associated signal transduction cascades, simple compounds like carbohydrates or lipids, secondary messengers, kinases themselves, phosphatases, which remove phosphate, the energy charge, and also stress. So mTORC is a major molecular switch, as I've suggested. It functions as a molecular relay to regulate a wide range of anabolic and, in fact, it inhibits catabolic processes. So I talked a lot about autophagy, about programmed cell death. This is the other side of the lens. This is the other side. This is when the sun's coming up. This is when everything's anabolic. Okay. So mTORC1 is a serine threonine protein kinase. That's what its actual molecular function is. That's how we can describe it. It promotes anabolism through phosphorylation of substrate proteins, including proteins like ribosomal S6 kinase 1, which we're going to meet up with here in this paper a little bit, and another really important protein, the eukaryotic initiation factor 4E binding protein, and that's involved in protein synthesis. Okay. So growth factors such as insulin will activate an enzyme complex called phosphorylated 1,3 kinase and the AKT pathway, and thereby that inhibits the tumor suppressor tuberosus sclerosis complex, also known as TSC1/2. And that, the reason I'm bringing that up is because insulin then is going to ultimately negatively regulate through that pathway. So if you remove that suppressor complex, you have a direct effect from insulin to mTORC. And that's, in fact, one of the ways that the experiments are done in this paper we're going to talk about. A loss of function mutation in the TSC1/2 leads to constitutive activation of the mTORC. So again, you can understand that if you remove that intermediary in the signal transduction cascade, you're going to basically have a constitutive mTORC going. Now, that when that happens, and it doesn't just happen in experimental design and in research projects, when that happens naturally in cells, it's often what causes genetic tumor syndromes and another really important series of diseases called lymphangioliomyomatosis, or LAM. Okay, so the hyperactivation of mTORC1 by an oncogenic acting, be a mutation, for example, P13 kinase AKT, or the RAS-ERK pathways, which I've talked about a lot in these video lectures, are also commonly observed in cancer. So this is a really important player in cancer research. All right, so on with the glasses to give you a little discussion about how complex mTORC is. These are various receptors that will bind to ligands, and those ligand receptor mediated process will all function through a series of enzymatic reactions. So for example, here's the epidermal growth factor, here's the MET receptor, here's the PDGF RAS complex, here's the insulin-like growth factor, here's just a direct amino acid consequence of receptor-mediated uptake. This particular slide is showing you gain of function versus loss of function. Those are the crimson spheres versus the light blue spheres in this whole pathway. And gain of function in what? Glioblastoma, and in either gain of function or loss of function in that cancer, in that brain cancer. So you see that what happens is you gain this function, you increase the amount of phosphorylation of this particular enzyme complex. That turns phosphorylated bisphosphate into phosphorylated triphosphate or trisphosphate. That's going to regulate the expression of a protein kinase, which is going to phosphorylate the AKT enzyme. The AKT enzyme, again, this is gain of function, okay? It's going to block this pathway. It's going to block that pathway, okay? So it's going to inhibit inhibition of mTORC. That's really important because the other thing it's going to do is work through RAS and through protein kinase C. Here's mTORC2, turning on protein kinase C. All of that, remember it's red, so it's gain of function in the cancer, going to move through to turn on mTORC1. So there's an mTORC2, that's just a different complex, but both of them are sensitive to the antibiotic rapamycin. That's what TORC means, target of rapamycin. So anyways, once you turn that on, okay, you ultimately lead to cell growth and proliferation. And that is linked to a whole suite of genes which are transcriptionally activated via this complex system in a thing like glioblastoma megaforma. And they move through a transcription factor called the SREBP. Now, that's the sterile response effector binding protein. The SREBP, what it does is it turns on the synthesis of lipids. So when you turn on the synthesis of lipids, both prenal and acyl, prenal lipids are things like cholesterol, And acelipids are, of course, fatty acid associated, and these can be phosphoglycerolipids or they can be phosphosphingolipids. So that means membrane biogenesis. So one of the key features of mTORC, because it's an anabolic pathway, now it's linked as a gain of function in a cancer like glioblastoma, is it turns on the synthesis of lipids. So it's a lipogenesis pathway. Why are lipids tuned up when you have a cancer? It's because you need more membrane. More membrane means more cell division occurs. When cell division occurs, you can make more cells, and that's the proliferation process that ultimately leads to tumorigenesis and maybe even metastasis. So that's what I'm trying to show you. There's a whole series of proteins here. And we've talked about a number of these in various talks. This is just a conglomerate sort of slide to get you to understand how mTORC can work through multiple signaling systems. But when it is turned on, one of the main features it will cause is the expression and the increase in the amount of fatty acid synthesis and cholesterologenesis enzymes in the cell. Now, here's a more simplified way of looking at it. Here's the mTORC1 system. Okay, you got amino acid transporters working through the lysosome. That's one system that we won't talk about today. You have the phosphorylated tris kinase pathways. There's the turning on of mTORC that we talked about already. Here's a direct activation of AKT. That little p means it's phosphorylated at that threonine residue. Here it is working through the tumor sclerosis factor. Now remember, that's going to block this pathway. So if you don't have this, this will be turned on, right? So mTORC being turned on then will turn on all this translational machinery, will turn on lipid and sterol synthesis, as we talked about in the last slide. And of course, whenever you make membranes, which is gearing up to make a new cell for cell division, vis-a-vis cell proliferation, vis-a-vis tumorigenesis, oncogenesis, you also, of course, need to do something else. You need to replicate the genome. So along with lipid biosynthesis tuned up in cancer cells, you also tune up or turn on the synthesis of purines and pyrimidines so you can make not only DNA for an entire increase in the genome so that you can go through cell division, but also a vast abundance of messenger RNAs so that you can make that cell anabolic so that it can get to the level of bioenergetics so that it can divide. All right. So I know that's, again, rather complicated, but I want you to see the real significance of mTORC. You don't need to worry so much about this TSC pathway, but just know that it's putting the brakes on it. And when the brakes aren't put on it, mTORC is fully activated. It's fully activated because of all of these signaling cascades, and these signaling cascades are ultimately, when you work through mTORC, turn on lipogenesis and turn on nucleic acid biosynthesis. Both of those are absolutely essential and universally important for cell proliferation. All right, so here's just a little bit more detail. mTORC increases the expression of lipogenic enzymes through the SREBP. That's basically a transcription factor, as I've said. But RNA splicing, processing, and stability have not been adequately examined as regulatory and pro-lipogenic activity. So, but we do know that mTORC is involved in that. Okay, so again, looking at these signaling pathways, you've got various kinds of proteins that are turned on, and those proteins, they basically are called SRs or HNRNPs. These are all enzyme complexes and RNA binding complexes which will alter RNA splicing patterns. There are also mutations which can alter splicing patterns. And those are just mean things like you get a problem with sequence, right? So there could be nucleotide excision, or there could be a nucleotide replication within the genome, and that's going to alter splicing. Because remember, the splicing is at junctions in the nascent RNA between introns and exons that originally came from DNA. And if you have an alteration because of mutation or because of specific binding of proteins at those intron-exon linkages at those sites where they come together, then you're going to have alteration of splicing patterns. All of that can cause deregulation of cellular processes. It can cause either cell death, so like the whole cell is starting to go south, it's going bad, so the cell kills itself, or it can proliferate, right? Other things that happen within these two poles is adhesion, differentiation, and changes in mobility and invasion, such as in metastasis. So you see how this is really important and how mTOR is going to be involved in this, right? Because mTOR is going to be associated with the transcription complex, and all of that seems to be associated with RNA splicing activity, okay, through these intermediaries. So post-transcriptional, pre- and co-translational control over nascent messenger RNAs can act in Byzantine via molecular mechanisms, including combinations of splicing, capping, polyadenylation, methylation, nuclear export, and stability. Now, all of these are mechanisms, molecular mechanisms, to get an RNA that's been transcribed faithfully, processed out of the nucleus, and available for protein translation, either on cytoplasmic ribosomes or in the ER polyribosome complexes, or RER. So I say they can act in Byzantine. That's my new term I like to use, because normally when you think about Byzantine, of course you know about the Byzantine Empire, the Eastern Roman Empire, but Byzantine also has a different connotation, and that is highly complex yet organized, right? Like over the top in terms of organization, over the tops in terms of hierarchical control, right? And that's what I mean by in Byzantine. So I just kind of invented that today for this talk, and I'm going to use it more often, probably the rest of my life. All right, and not just professionally. All right, so those processes are regulated in part by various RNA binding proteins, okay? And that's what we're talking about here. So you have serine arginine, that's the SR, it's where it comes from, proteins, and you have this HNRNP, which are heterogeneous nuclear ribonuclear proteins. Those work in conjunction to deal with the processing of the nascent messenger RNA at the level of clipping out introns, fusing together exons, and making that messenger RNA, that processed messenger RNA, now available for synthesis of proteins, that is through the translational machinery. Okay. So that's the important thing to keep in mind here. So the SR proteins are encoded by serine and arginine-rich splicing factor gene family. Those are called the SRFs, or the SRSFs. And they're composed of an RNA recognition motif called an RRM and an arginine-serine repeat domain, or an RS domain. Okay, so this is getting into the fine molecularity of what SR protein gene structures look like, and also the various functional and structural domains. I put that in there for those of you that like that detail. I like the detail, and I know some folks, well, you can just tune that out when I talk about it. Anyways, what these SR proteins do is catalyze, right? Because they're enzymes, the messenger RNA processing by directly binding to exons, okay, that is binding to the nucleotide sequence, in pre-mRNA, and recruiting proteins such as the small nuclear ribonuclear proteins, the snRNPs. So this is starting the spliceosome project that ultimately leads to the mature RNAs. So the activity of SR proteins can be regulated via phosphorylation. So you can regulate the activity of these proteins that are going to be involved in the processing of the nascent RNA via adding a phosphate to them. And you add the phosphate to the RS domain by an enzyme called an SR protein kinase, which are going to play a major role here in this talk. And these are called SRPKs. Gee, not hard to understand where you got that acronym from. Serine arginine protein kinases. You get it? Right. They alter then the protein conformation when you phosphorylate those enzymes. And then when you add that phosphate, change the conformation of the protein, you're going to change subcellular localization. And you're going to also change the protein-protein interactions of that now nascent phosphoprotein. And all of that is going to play a major role in the talk we're doing today. Okay, so that's really important for you to understand. Subcellular localization is important because if you're talking about anything that's going to involve the Byzantine control over gene expression, you can talk about everything that's happened in the cytoplasm, but what else you got to worry about is where that RNA was first made. Where's the RNA first made? That is correct, unseen voice and unheard voice. It is happening in the nucleus. So for these proteins to work, they have to end up in the nucleus. Most useful is the fact that when you phosphorylate these proteins, they end up there. Okay, let's continue. This is from a paper in Genome, published in 2009. SR proteins, that's the green one here, bound to an exon splicing enhancer element, that's the ESC, so there's between the exon and another exon, associated with all these other proteins here, which you don't need to know, but there is an snRNP, ribonuclear protein. Okay, they interact with the splicing factors UTAF, right? Bound to an upstream 3', that's 3' from the site where you're going to get this excision, splice site, and the U1 snRNP bound to the downstream 5' splice site. So this is all part and parcel of how we work with messenger RNA. And all this processing is, if you had a recent course in molecular genetics. I'm sure you'll be aware of this. If you haven't, which I assume most of you haven't, don't worry about it. I'm just showing you that all these proteins are potential targets. We're not going to look at these right now. We're just going to emphasize this SR protein. Remember, that's what the cell paper is going to be talking about. Now, exon-independent function of SR proteins, there's also that. They have two exon-independent functions. SR proteins facilitate splice site pairing by simultaneously interacting with the U1 snRNP and the UTAR AF across the intron complex, okay? And they also assist in recruiting that whole complex to get it onto the nation transcript so you can start doing this processing. Okay, I know that's a lot of detail, but I wanted to make sure you knew that there is that kind of detail in splicing. So splicing repression is mediated when SR proteins associated with intron sequences close to the splice site. Now, when that happens there, the recruitment of the spliceosomal components then doesn't occur. So the SR protein itself, rather than interacting first with all these proteins to make the complex to carry out the spliceosomal project, if SR protein binds first, it stabilizes that intron-containing messenger RNA, so you don't get the intron removed. So that stabilization of the messenger RNA basically means you don't get expression of the gene, because you haven't made then the formal, mature messenger RNA, means you spliced out the introns. If you haven't spliced out the introns, that messenger RNA is going to be doomed. It's going to be broken down because it cannot be translated. So that's the reason I showed you all this detail. So SR protein then, it's involved in putting everything together so you get a spliceosomal project and you make that mature RNA so you can get protein synthesis, protein translation, protein expression. All those are synonyms for this talk. But dig it, it can also bind directly to certain regions right onto the intron exon boundaries. And when it does it blocks everything else. You see, so everything has one more level of complexity than you might guess. Actually, only one more usually, right? But that then turns 1 to 2 and 2 into 4, and you get the logarithmic increase in the level of complexity, which is why I call it Byzantine. Data from this research will suggest that the mTORC1 dependent regulation of lipid metabolism involves a unique effector kinase. Remember, this is an SR type kinase, so it's going to be SRPK isoform 2. So the mTORC1 dependent kinase, CRPK2, effectively will regulate the expression of lipogenic enzymes, and it's going to do it at the level of ultimate splicing. So we're talking about expression of gene, but we're not talking about transcription. We're talking about post-transcriptional modifications leading ultimately to translation, right? So now what's really interesting is nutrients and growth factors act upstream of this whole process, okay? So nutrients then, say, turning on insulin secretion activating mTORC, and then the rest of it follows through as a sequence of events leading to control at the spliceosome level. So this is all really important. That's why it's in the journal Cell, premier journal, because SRPK then surfaces, because of papers like we're reading here that are only a month old, as potential therapeutic targets for mTORC1 driven carcinogenesis in chronic metabolic disease. So there's two different major diseases here which cause a lot of death in humans. Cancer and the whole series of metabolic diseases associated, for example, with obesity, right? Such as metabolic syndrome and also type 2 diabetes, right? Because insulin's going to be involved in this. So mTORC phosphorylation, substrate conservation, and source of monoclonal antibody detection for the Western blots. Now it's basically, this is a materials and methods sort of slide. So I went through the end of the introduction. I'm showing you, here is that gene complex. And this SRPK2 is how they can serve between humans and zebrafish. You can see here, they all have this very conserved serine sequence event here, right here, serine 495 to serine 497, and all of them are these two serines which are going to be phosphorylated and therefore control the movement of this kinase into the nucleus, where it's going to be affecting the initial project of causing that spliceosome preparation so that the messenger RNA can ultimately leave the nucleus and be available for maturation so that it can be synthesized, used to synthesize polypeptide. Now notice that all of these different, quite diverse animals all have that really tight sequence similarity. That's because that's a really hot site for adding phosphate, those two series. So that's why I'm showing this to you. Now, what you're going to find out is that in the paper that we're going to look at right now, the one that I told you we're going to look at, the Cell paper, is that monoclonal antibodies have been raised not just to the kinase itself, not just to SRPK2, but to the phosphoforms. Now, that's important because it's the phosphoform that is the functional enzyme. So if you don't have those phosphates via mTORC, you see how this is working upstream via signaling, then we're not talking about anything. We're not talking about any regulations. You got to have those two serines phosphorylated. So you got to have antibodies to be able to detect them. And they got them. All right. So here's some data. This is an immunoblot. So basically an immunoblot is at the protein level, right? And the immunoblot analysis in a particular cell line called HEC293E, I've actually worked with these cells in the laboratory myself. Very good mammalian cell line. And these are transfected with, a couple of molecular tools. A small interfering RNA, and that small interfering RNA targets the protein kinase, the serine arginine protein kinase, which remember is activated via the mTORC1 complex, okay? So you've got an siRNA, and when you have an interfering RNA, what that's going to do is it's going to block the activity at the RNA level of this kinase, okay? Now you also have rapamycin. Now rapamycin isn't functioning at the level of the protein kinase. What does rapamycin do? Remember what mTORC is? Molecular target of rapamycin. What happens when you add the antibiotic rapamycin? You shut off the mTORC, okay, because it's an antibiotic and the target of which is mTORC. So add rapamycin, it's going to act like the small interfering RNA, right? If these two are linked, if mTORC and SPRK are linked. Get that? So two ways of doing it. You're going to activate the whole system via insulins. You're going to add insulin, 100 nanomolar. And the way these cells that were first depleted of other cofactors and growth factors, so that when you add insulin, you get an immediate effect. So some of the details of how the experiments are done. And you do a 2-hour time course after you add the insulin, and it's with or without this antibiotic rapamycin. You see, you don't need much for 20 nanomolar because it's very specific for binding mTOR. And you get a treatment of 30 minutes. Okay. Now, dig what happens here. So this is if you add insulin. This is no. Notice that if you don't add insulin, you don't get any phosphorylation because the antibody is to this phosphorylated serine residue. Boom. You add insulin, you get an increase with using the antibody that detects the phosphoprotein at the 494 level, the serine 494. You get an increase at the immunoblot level of that protein. That means you got more of it, right? But if you add rapamycin, boom, you lose the increase. That tells you two things. That tells you that insulin causes the phosphorylation of that site. It also tells you that it's mTOR dependent, because when you knock down mTOR, you don't get it. See, so mTOR's got to be the intermediary between insulin binding to its tyrosine kinase receptor, and then mTOR actually causing the SRPK to become phosphorylated, because that's what mTOR is. Remember, itself is a serine kinase. Now, also dig that when you add both of them together, okay, you get a decrease, right? You get a decrease in its activity, and this is shown twice here. The numbers are just showing you fold changes. So there's a threefold increase. This is less than half of what that was in non-stimulated. And then now notice that the interfering RNA also knocks down from the non-affected, but also from the enhanced via insulin activity. So what you're seeing here is really important. Oh, by the way, these are this control here. They added a small interfering RNA, but it was not associated with the nucleotides that are part of that gene, right? So these are non-specific, and so you see that just adding it's a control. Adding just small interfering RNA had no effect on any of this. When you add the specific one, you kick down the phosphorylated form of that protein. That's because this is a small interfering RNA system blocking the whole level of protein, right? And that's what you see down here. There's no effect on the non-phosphorylated form when you just look at the effect of this antibody, right? Because this is this SRPK2, not those ones that are phosphorylated, just the total amount of protein. So what we're looking at is a very fine-tuned control. So until we add these antibodies to those phosphoforms of this protein, we would be looking at these protein plots and say, well, it doesn't seem like there's any activity with insulin or rapamycin. Gee, insulin must not have any effect on this kinase cascade system, but we know is associated with spliceosomal alteration of gene expression. But when you dig down into the molecularity of it and you get the antibodies to the phosphoforms, you find out, yeah, that's true. Total amount of protein doesn't seem to be affected very much. But wow, when you look at that interfering RNA for that, you find out that indeed it does block that, because the interfering RNA works at the molecular level of just the holoenzyme, right? You're not going to make the protein. But look also that it affects directly, right? Yep, the phosphoproteins in it better. Right? Okay, so that's here. No effect on non-phosphorylated basal SPRK2 protein levels with rapamycin, but the abolition of protein using just the sRNA. So see, that also tells you that insulin's not doing much here. It's at the level of how much protein is there total. It's only affecting the phosphoprotein level, okay, both forms. And you also notice that rapamycin is not doing anything really to alter the expression of the gene. You only talk about the alteration of the expression of the phosphoproteins. That's what this is all about. Okay. Now, in growth factor independent mTORC constitutively active cancer cell lines, the same effect is observed. So these are when you knock out that tumor sclerosis complex. Remember that was the intermediary that's going to block, right? That's going to block the mTOR activity. So that's the normal putting the brakes on in vivo. So you've eliminated that, and this was done by gene ablation via gene knockout, that is homologous recombination mediated gene knockout. These are cell lines that come from angiomyolipomas. Okay, so this is that disease we talked about, right? And these are bladder cancer lines, and these are breast cancer lines. So 3 different forms of cancer, right? And this one doesn't utilize that method. This one here is looking at just the P13 mutations in the P13 kinase and the ERK kinase. Okay? So those mutations in those kinases phenocopy the TSC1, which is normally not found in this breast cancer cell line. That's why we're not using it. Okay. So look here. You're looking at the phosphoforms. DMSO is just a carrier, right? Dimethyl sulfoxide. Yeah, the activity is 1. You add rapamycin, boom. You get it down to about 1/3 with both different phosphoforms in this cell line. And this is now constitutive mTORC. Why is constitutive important? You don't need to turn it on with insulin. You see? Constitutive activity. Same thing here. Phosphoforms are kicked down to about 1/3. Both forms. This was actually kicked down to what, about 1/5 or 1/6 in this particular phosphoform, looking at the bladder cancer line. And you get the same effect for that phosphoform in breast cancer. And you also get a decrease in this phosphoform in breast cancer. But look at, there's a big difference there. And the difference is in the level of hierarchical phosphorylation of those 2 different sites. There's also a slight difference in bladder cancer versus breast cancer versus this angiomyolipoma. But it's just showing you they all trend, okay? So that's telling you that you're working through that pathway via mTORC. Now, data that I'm not showing here, because there's so much data in this paper, which is indicative of Cell papers, is, and all the figures too are what are going to be summarized right here. That whole series of figures and those research, those experiments demonstrated that S6 kinase 1 directly phosphorylates SRPK2 downstream of mTORC signaling. Remember I told you that serine kinase, that serine 6 kinase was really important. You saw those initial introductory cartoon slides. Well, guess what? That's what they had to prove here, and they did by doing all those experiments. Adding inhibitors of this kinase, adding knockouts of the kinase, the whole thing you just saw with going directly after the SPRK. They just showed that this is an intermediary in the system. Okay, that I think was important to show you all the data, because that's really all it is, just filling in the blanks in the signal transduction cascade. Now, phosphorylation of the SRPK2 by mTORC signaling affords this nuclear localization. So once mTORC phosphorylates that enzyme, it makes it to the nucleus. Translocates the nucleus, it phosphorylates the downstream SR proteins. Remember, those are the ones that are involved directly in the spliceosome situation. Constitutive SRP kinase 2 was localized in the cytoplasm. When you treat with insulin, it goes to the nucleus. Nuclear accumulation is correlated with phosphorylation of this really important serine arginine protein that's going to be affecting splicing, and that was increased by insulin. So this puts together the whole package. Insulin, mTOR-mediated phosphorylation of SRPK2, movement into the nucleus, phosphorylating its target, the SR protein. All of that is all linked together. inhibition, and we know this because inhibition of the mTORC activity by rapamycin induced by cytoplasmic retention, induced a cytoplasmic retention. So you never get the protein out of the cytoplasm if you inhibit mTORC. That's because it's working through the SR6 kinase, phosphorylating that protein, that is the SRPK, and allowing that protein, once phosphorylated, to be targeted to the nucleus. Okay? All because of mTORC. And rapamycin knocks that back. So results show that insulin, mTORC-C1 signaling promotes nuclear localization. All right. So we talk about how splicing affects gene expressions at the global level and then at the specific level. So here's DMSO, the carrier, versus rapamycin. So you see that 344 proteins are up, okay, when you compare DMSO to rapamycin. When you use a small hairpin, basically a small hairpin which is going to block the SRPK versus a small hairpin for just the green fluorescent protein. So that's a control, and you can measure, and they measure the green fluorescent protein so they can knock back green fluorescent protein. Look at that go away. At the same time, they have the small short hairpin RNAs to inactivate the SRPK. So they did that via cell sorting, cell biology type of research. Really beautiful slides. I'm not showing you that. 233 genes up versus 175 down, showing you when you interfere with that protein, you get an alteration in gene expression. When you add the two, Venn diagram-like, and you look at both rapamycin and whether or not you inhibit that enzyme, this is telling you this is at the mTOR level and specifically at the SRPK level. 42 proteins come up and only 21 go down. So those are the ones you want to be interested in, probably. So SR proteins are implicated in messenger RNA biogenesis, including, as we've said at the beginning, transcription, splicing, export, translation, and stability. So here's the splicing index. Again, just superimpose this data onto that. So DMSO, the carrier, versus rapamycin, because rapamycin has to be put in dimethyl sulfoxide for it to work. You see here that you get a difference in genes. And the same thing with the GFP versus the small hairpin. Inhibiting that particular pathway, you see a difference in genes. And when you combine the two, that's looking at rapamycin and directly at its downstream phosphorylated product, you get some 500 genes, which look like they are affected at the splicing level. Okay? So this is just looking at total level of protein, right? And this is looking at the splicing level. So whole transcriptome, this is how this is done. Marker RNA analysis of gene expression and exon intron utilization, because you're looking at splicing products here. That's why it's called a splicing index. Demonstrated that SRPK2 does indeed regulate gene expression and splicing via versus the DMSO vehicle, and that rapamycin phenocopies some of that. All right. So now we're getting finally down to what are the proteins that are being affected. SRPK2 regulates the expression of lipid biosynthetic enzymes via mTOR. So here's a fold decrease. You see a decrease in some of these proteins here that look like they're most decreased here are going to be, now these are proteins associated with lipid biosynthesis, as we'll see soon. So rapamycin is going to cause a decrease in some of the activity of some of these enzymes, right? Likewise, when you use the short hairpin for the SRPK2, another way of blocking it at its activity, some of the same proteins show up. You get a fold decrease. This is just another method showing you the specific and discrete proteins that are mediated via a decrease in either the SRPK activity, which you know is going to be associated with the nucleus, and you know it's going to be associated with phosphorylating the SR protein, which is going to then link to the removal of the introns, right? So that's the important data point here. All right. Now, both prenyl, if you look at what genes were involved there, both prenyl and acid lipid metabolism are controlled via this pathway. And it's all going down with splicing reactions in the nucleus, because we know that from the previous work. So here are some of the enzymes involved. Okay, so this is the ATP citrate lyase. That's the enzyme that takes citrate, which was synthesized in the mitochondria, translocated out of the mitochondria in the cytoplasm, and then cleaved into oxaloacetic acid and acetyl-CoA. That's the beginning of lipogenesis. Once you cleave citrate back to OAA and acetyl-CoA can be used as precursor substrate for prenyl lipid synthesis, that's cholesterologenesis, and acyl lipid synthesis, that's fatty acid synthesis, which of course includes things like triacylglycerol, but also the phosphoglycerol lipids and of course the sphingolipids. All right. And so Fatty acid synthesis protein is also affected by this, as is steroid desaturases, right, which is one of the enzymes which turns on the synthesis of oleic acid. Right? So it's just showing you the various genes and the fact that rapamycin, as compared to DMSO, knocks that back, as does taurin. That's a specific inhibitor of the complex. So rapamycin is the antibiotic. Taurin is a peptide inhibitor of the mTORC pathway, showing you a lot of significance for these particular genes. There's HMG-CoA synthase. This is an enzyme involved in acetyl-CoA synthesis, right? And various enzymes involved in either lipogenesis in general. Okay, so inhibition of these pathways, you would argue, would block them de novo lipogenesis, and because you see it is, and that suggests a possible protection in tumors. All right. So here's small interfering, non-specific small interfering molecules. You're looking at the amount of acetate incorporated into fatty acids. You see here that when you use this specific small interfering RNA, you get a much bigger decrease in the amount of acetate that is incorporated into cholesterol. This is the amount of acetate incorporated into fatty acid. Now, why is this leaky? It's because obviously more things are going on than just this, but you see very significant decrease in the cholesterol level, and that's what's important here, okay? And you see that when you have the nonspecific nucleotide, the small interfering, no effect on fatty acid incorporation, but when you add the specific, you do get it. Likewise with cholesterol. So this is a radioactive uptake assay using just acetate, and both fatty acid decreases because of this specific interfering RNA, and cholesterol decreases. Same thing here, phenocopying it with rapamycin or the mTOR pathway inhibitor, the taurin-1. Again, the amount of acids incorporated into fatty acids, either with rapamycin or taurin, blocking mTORC, bad compared to the carrier molecule. You get great decreases in the amount of fatty acid synthesis and also cholesterol synthesis, way lower levels. Okay. Now, that was done at the C14 level. Now we're looking at heavy isotope data. This is C13 using mass spec. So it's just a cell paper. They did everything, and they did it twice. Thank you. And I love this paper. I love this data point because it's looking at palmitic acid, stearic acid, and oleic acid. De novo synthesis, looking at the amount of C13 incorporation into these fatty acids, doing mass spec bombardment to look at these paired ions. And you see that palmitates kick down by looking at the specific inhibition, this SRPIN, that's an inhibitor of that enzyme. Every time you do that, you decrease the amount of incorporation of C13 into these three fatty acids. So you're knocking down at the transcriptional level, the post-transcriptional level via the spliceosome complex, those enzymes involved in fatty acid synthesis desaturation. And indeed, you find that plays out, that translates into a decrease in palmitate stearate, and there's the first desaturase complex. All right. Again, now we take it all the way to proof of concept in rodent studies. This is a xenograft of tumors. How much it grows, normalized to week 1, 4 weeks after you've xenografted that into the mouse. There's the control, and there is the knockout. There's a knockout of that enzyme. Look at that. Even though you've xenografted those tumors, if you knock out that particular isoform of that kinase, which we know is involved in mTORC-mediated control of spliceosome, alteration of gene expression of fatty acid synthesis and cholesterol genesis, look at that. You really get a knockdown in tumor growth. That's really important. This is the LAM system we were looking at, okay? All right. Likewise, same kind of thing going on here in these other cancers. There's a xenograft of 2 different types of tumor lineages. Once again, this is looking at inhibitors of that serine kinase. You inhibit that kinase, you do not get good tumor growth in these other cancer lines. All right. The dialectic now, almost over. Insulin-induced mTORC-facilitated phosphorylation of SRPK2, which ultimately increases messenger RNA stability of lipogenic genes by promoting efficient intron splicing. So that is the premise. We know that happens. Now, we also know that rapamycin, or the interfering RNA, interfering RNA should be IS, not SI, sorry, or the inhibitor of that enzyme caused intron retention. That's destabilized that messenger RNA. You got a degradation of that nation RNA, and that RNA happened to be specific for lipogenic genes. Fatty acid synthesis, fatty acid desaturation, HMG-CoA synthase, all those enzymes involved in either fatty acid synthesis, acyl synthesis, or the prenolipid synthesis. All nicely linked. So that's the next step in our dialectic. And then the conclusion, therefore, blockade of the mTOR pathway will tank lipogenesis, and that's linked to a decrease in tumor size, as we just saw with the proof of concept data, via post-transcriptional mechanisms. Pretty neat paper. And here's where they put everything together. And this is from the authors themselves from the paper. So mTORC, working through S6 kinase, will cause the phosphorylation of that serine residue on SRPK2. We didn't talk about it, but their data also showed that this other kinase will also phosphorylate this enzyme. Once you phosphorylate SRPK, it phosphorylates in the nucleus. Now we're in the nucleus because it gets translocated there, remember, because of this phosphorylation due to it, all mTORC mediated. It's going to link up with this UI70K. Remember, that's the protein involved in the spliceosome around that RNA, that intron exon junction. And when all that's working collectively and correctly through SRPK, you then take that RNA and you splice it and you get increased in stability, you get increase in fatty acid synthesis, prenolipid synthesis. However, if you cause intron retention by inhibiting either mTORC or inhibiting the SRPK, then you don't get that. You get intron retention, you decrease stability, and you don't get more lipid biosynthetic enzymes. And remember, you also need to translocate this binding protein because that's part of the chromatin remodeling, right? Opening up the enhancer element and the promoter element, because these are all responsive through the sterile receptive element binding protein. And that also gets processed because of this whole pathway, something that the paper talked about, but I just now am going to mention to you. So that's basically the story. It looks like we have a very positive target now to block tumorigenesis by blocking the stability of RNA that codes for lipogenic enzymes. And we're looking at doing it by inhibiting SRPK2, or perhaps by blocking mTORC, maybe even with something simple like rapamycin. So there it is, the entire paper. Hopefully that was interesting to you. It was certainly interesting for me to read and to learn about, because I am a lipid biochemist, so I like to know how lipid biogenesis is regulated. And I'm looking in now in a whole different light, because I'm seeing that at the splicing level via mTORC, one of the anabolic activities of mTORC is to positively regulate splicing of fatty acid synthesis and fatty acid desaturation and prenolipid synthesis, that is cholesterologenesis, all at the level of splicing, all initially at the level of the nucleus. I think that's really fascinating. Just yet another level of complexity, another Byzantine understanding of how beautifully complex the cell is. All right, so I'm going to end here. That's me. I'm Dr. Dan Guerra. That's my email address. Please contact me about this lecture or any other ones you see that you did or didn't like, and let me know. Give me feedback, please. I'd love to hear it. That's my Facebook page, where this particular video will be uploaded very soon, and you can find it there with all my other videos. It's also on my Twitter page, but I don't have that Twitter page site, but you can find it by going to the Facebook. Here's our website. Here's our company. There's our email address to our company. There's our company website. Please go visit it. Remember what we are. We are scientists verifying published evidence in medical biosciences. What I gave you here was an example of that kind of activity that we do. When I go in and I take a look at that, which is looking at one paper, right? So, of course, if I'm working for you, I'm going to be looking at whatever topic you have, and I'm going to be looking at papers like we just did there. And by examining that paper and maybe 20 or 30 other ones, depending on how deeply we get into it, I can help explain something that's going on with you, say, in the laboratory, going on with you at the clinic when you're looking at different drug-drug interactions for therapies. And also, if you're a patient and you're interested in the course of events that are going to be leading to, let's say you've been diagnosed with cancer or metabolic disease or just with obesity, well, how the drug's going to function. I can tell you that a lot of the genes we talked about today and a lot of the proteins that are involved in those signal transduction cascades are actually targets for those diseases. So this just gives you a detail of that. And of course, for your specific situation, I'd be glad to talk and give you a complete synthesis of what the literature says. Again, we work with physicians, with their clients, with their patients, and we also work directly with physicians and also with other professionals such as lawyers. And of course, just people that are interested in knowing more about any of the science that I read every day. So with that, I'd like to say goodbye. And I like to say my sign-off very specifically by saying goodbye for now.